title of my talk is Great Ideas of Biology. Notice it's not the great ideas of biology, because I like a little bit of sort of fluidity there, but it's my choice of what I think are great ideas that perhaps we should be thinking about in biology. Because on the whole, biologists tend not to talk about great ideas and grand theories. In this respect, we differ from the physicists who love to talk about great ideas and grand theories. Biologists tend to be more comfortable with details, with particulars. We like to list the numbers of species in a habitat, or if we were a 19th century biologist, we'd count the numbers of hairs, perhaps on a beetle's leg. Today we would sequence genes or determine all the RNA and proteins that are in a cell, much the same thing as counting the numbers of hairs on a beetle's leg, just a bit more high-tech. And because of that interest in details and particulars, which are very important in biology, we tend not to think about the great ideas. But biology does have great ideas, and I'm going to talk tonight about 4 of these that I think many biologists would agree are important. And I want to point in the direction of a 5th, which is yet to be fully formulated, but one I'd like to share with you as one I think will be of importance in the future. Now, I'm going to start with the first idea, and that is the idea of the cell as the basic unit of life. And I should say that in my discussions of the different topics, I'm going to talk about the history of the idea and some of the philosophy behind it, and emphasize some of the connections. And so I want to sort of give you a sense of where the idea came from. Now, this first idea, easily stated, all life is composed of cells, and the cell is the simplest unit exhibiting the characteristics of life. You can view the cell as life's atom, if you like, the basic unit of life. Now, this idea started in the 17th century, and it took around 200 years before it was fully developed. And as is often the case in science, it was technology which begat the discovery. And in this case, it was the technology of the microscope that was discovered or put together in Holland at the beginning of the 17th century, and was used by Robert Hooke in the 1660s to look at plant material, in fact a lot of different biological material. Now, Robert Hooke was the experimentalist for the Royal Society, which had been founded in 1660 as a club for those interested in the natural sciences. But this was really very much a gentleman's club, and I think they hired Robert Hooke to be their experimentalist in the first instance. I suspect they didn't actually want to dirty their hands with actual experiments, but hired somebody who would actually do that, but would demonstrate the experiments and allow a proper discussion of what the experiments meant. So the experimentalist for the Royal Society, Robert Hooke, did a lot of different experiments and studies from biology through to physics. He had a notorious row with Isaac Newton, who then tried to destroy all trace of him after Hooke died. Don't get the idea scientists behave badly to each other just today. They've been doing it for hundreds of years. And what Hooke did was to take a thin razor and make a slice through plant material, in this case cork, and looked at it under a microscope. And what he saw was the image you see. This is what he drew, the black and white image. And it looked like, to him, like a honeycomb of cells. And he called these square boxes here cells. Some people say it reminded him of a plan of a monastery, and he thought of these as monk cells as well. I'm not quite sure if that's true. On the right, we have a modern scanning electron micrograph of the same material. So this was the first observation and the first description and naming of the cell in 1665. Then, having made that discovery, it was quickly followed by many others. That's also quite a common feature in science. Once one person has discovered something, the whole world discovers it. And within a few years, this is 1665, you saw images like this. This is 1682, so only a few years later. And it's a section of a vine plant drawn by Nehemiah Grew, who was a fellow of the Royal Society as well, and an MD. We have I put a modern picture up there on the right. And this one over here would be of a quality that you would have seen in a 20th century textbook on plant anatomy. It's completely extraordinary. Of course, plants are easy to preserve and to cut and to look under the microscope, but it is quite extraordinary, the detail that Grew drew here. I have the book from which this image was taken, and I have another book from 1672 by Nehemiah Grew, which also has pictures of cells, and I believe it's the second book that describes cells. I can't afford the Robert Hooke one. That cost you about 100,000. My one, only 7 years later, cost about 200, so much cheaper. At this time also, single-celled organisms were discovered by LEEUWENHOEK, and there was other work by Malpighi. LEEUWENHOEK described single cells. Here's a picture in a letter he sent to the Royal Society. And what he did is he took a little scraping from between his teeth and put it under, in fact, a single-lensed microscope, which had less aberrations than the complex microscope. And he got extraordinary magnification, so that these are bacteria. If any of you took a scraping between your teeth, I'm afraid to say you would see the same thing. And rather charmingly, he drew what he saw. If you look at figure B, you'll see that clearly that bacterium did a loop-the-loop under the slide as he was looking at it. And LEEUWENHOEK sent about 1,000 letters to the Royal Society at this time, describing all sorts of things, but particularly pictures related to cells. He was a draper in Delft. He was from the Netherlands, and he wasn't particularly scholarly. He couldn't write in Latin, so it was sent in Dutch to the Society. And we do have a rather poor likeness of him, but I like to think that there was a painting done of him, because LEEUWENHOEK knew the painter Johann Vermeer, and Vermeer died, and LEEUWENHOEK was appointed the executor of his will. And unusually, Vermeer did 2 paintings of a scientist. And I do not actually know of any sort of unknown pictures of scientists of the time. Obviously, if you were an aristocrat like Boyle, you were painted. But this was an unknown scientist, a geographer and an astronomer, one in the Louvre and one elsewhere. This is the one from the Louvre. And I just like to think that this actually might be LEEUWENHOEK. But there isn't a scrap of evidence, as far as I'm aware, for that speculation. But since I'm a fan of Vermeer, it gives me a good excuse to show you a picture of Vermeer. So this meant by the beginning of the 18th century, cells have been discovered. They've been seen in plants. They were also seen in animals. In blood cells were discovered first, and single-celled organisms have been discovered. The next 100 years was slow. Microscopes improved, methods of fixation and so on improved. There was some philosophical speculations that complex animals and plants like ourselves should be considered as an assemblage of single-celled organisms like bacteria. And this was being talked about by German scientists at the end of the 18th century. And by the beginning of the 19th century, the cell doctrine was born by a book by Theodor Schwann and Schleiden. This followed close on the heels of Dalton, I should say, describing atoms as the basic unit of matter. And Schleiden, who was a botanist, and Schwann, who was a zoologist, came to the conclusion, in German, of course, in a book of 1839, we have seen that all organisms are composed of essentially like parts, namely of cells. And that is half the cell theory. It's the half which says cells are the basic structural unit of life. But of course, cells are more than that. Cells are also the basic functional unit of life, and that was being developed in parallel and found its clearest expression by a pathologist, one before Antony, Rudolf Virchow, in fact the founder of pathology. And in Germany again, Virchow wrote in 1858 in his book on pathology, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. So that is the statement that the cell is also the basic functional unit of life. Now, this conclusion, these two conclusions here, are a major milestone in biology. I actually don't think they get the airtime they deserve. Some of the other ideas you will hear about more, but this was a major milestone that the cell is the basic unit of life. And the cell, of course, underpins all reproduction and development. We know from high-quality photomicrographs much about the structure of cells. Because I was in Rockefeller, I mentioned some of the Nobel laureates who founded cell biology there. But the cell undergoes, it's the basis for reproduction and development. Cell division and cleavage of a single cell produces the embryo, and it's very important for understanding reproduction and development in all living things. If I haven't persuaded you of the importance of the cell in biology, then I'm going to show you this image here. This is an image of a mammalian cell, of an egg being fertilized by a number of sperm. And I'm going to remind all of you in this audience that you once, every one of you, looked like that. This is a single cell. You were all once such a single cell. And if I haven't persuaded you that it's interesting now, then I should stop now. So every one of you looked like this. And not only that, it continues to be crucial in our thinking about biology. It's, of course, the basic unit of life, as I've emphasized. It underpins all reproduction and development. And nearly every week, when you open the newspaper, you will read something, for example, about stem cells and the importance that they may have to play in development and in thinking about disease, including cancer. So cells and cell biology is a crucial part of all biology, and that's my first great idea, the cell as the basic unit of life. The second idea I want to talk about is the gene as the basis for heredity. Now, from ancient times, it was observed that particular characteristics are passed down from generation to generation, from parents to offspring. And speculations were made about what was the basis of that heredity. And this was a problem that was being tackled in the 19th century by particularly plant breeders trying to improve crop plants. And whilst they were doing that, they were thinking about the crosses they were doing between different plant varieties with different characters. They were thinking about the problem of how characters were inherited from one generation to the next. Now, this was quite complicated. Quite a number of workers observed that on occasions characters became blended. So if you had two plants, you crossed them together and they crossed. The offspring sometimes in certain characters were intermediate. Sometimes one character would disappear and it would be dominated by the other and then would reappear in later generations. And this is all very, very confusing. And this was a problem that was solved by the great gardening monk, as I called him, the originator of genetics, Gregor Mendel. And Mendel worked in Brno, in what is now the Czech Republic, then part of the Austro-Hungarian Empire. And he sort of belonged to what I can only describe as a sort of order of scientific monks. We don't sort of realize how the landscape of science has changed in in modern times. But in the mid-19th century, because he was working in the 1860s, there were certain monasteries which would specialize in different areas, and the monastery that he joined was particularly interested in science, and they did scientific research. He himself was trained as a physicist, and he was supposed to be trained to a level where he could go out and be a teacher. He actually wasn't a very good physicist. He didn't do terribly well in his university training. He failed some of his examinations. But he was very curious, and he did a certain amount of physical research. I've seen his research in physics, I should say. I've seen his telescope. He made little maps of the moon, for example. And he also was interested in meteorology and used a barometer to show how air pressure changed when you had a violent storm passing through, because it drops. And he turned his skill set that I think he learnt with his training in physics towards plant breeding. And he had quite good scientific facilities in the monastery. I know it quite well. This is a picture of his garden. It's still there. And adjacent to this was his greenhouse. And his greenhouse was quite a major facility. I mean, it certainly would be stretched from here to the end of the wall and up to about the third row. So this would have taken quite a big grant to the research councils to produce a facility of this type. Now, what he did was he argued that to sort out what was happening here, you had to approach the problem rather systematically, because biology at the time was dominated by a more a natural history ethos of describing what you saw, but in a qualitative sort of way. But because of his physical training, he, I think, thought that what you had to do was not only very accurately catalogue the types of plants that you were seeing, but also count the numbers, which I think is not something that most biologists would have done. But I'll say something about that in a moment. And he approached his problem systematically. And there's several reasons, I think, for his success apart from this approach. The first thing he did was he chose material that he could understand. And this is quite interesting. He did a number of trial experiments with a variety of different plants, 5 or 6 different species. And most of them were just very confusing, just like the other plant breeders had found. But instead of actually devoting his time to studying the confusing material, he just moved on to other plants where he felt he could make more sense. And you could almost say he was cheating. He was sort of moving around till he found something he could make sense of. But actually this is very, very important, because when you're dealing with complex phenomena like in biology, if you choose the right material, you can sometimes make sense of it. And once you've made sense of it, you can then apply what you've learnt to more complex systems, and you can make sense of those too. He chose the P, and he chose the P because it had clear characters, like tall plants, short plants. I think I've got some pictures here, tall plants, short plants, different coloured flowers, different coloured seeds and the like, which he could easily categorise. And in his trial experiments, he noticed that there were simple ratios emerging from the different crosses he took. And this was because fewer genes were involved in determining the characters that he was interested in, and in some of the other plants he was looking at where there were more genes, the whole thing gets very complicated. The simple ratios that emerge disappear. So he chose material he could understand, important in science. Secondly, he recorded accurately the types of plants he saw, and he counted them, as I've already mentioned, because I think he was a physicist, and also because he was trained as a physicist, and this was unusual for biologists too at that time. He generated a theory or law that he felt could explain his data, and then went on to test it. And that's, in a way, again, if you're a natural historian, you tend to describe and not generate these theories. And what he observed was that in the first generation after a cross between 2 different sorts of plants, the same species but different characters, often one character would dominate, but the other character re-emerged in the second generation when you cross them again. And often appeared in distinct ratios. Now, I'm not going to do a 101 genetics to you here tonight. I expect you're glad about that. But some of you will certainly remember ratios like 3:1 and 1:1 and 1:1 and so on. And the famous ratio is 3:1. And he thought about this and came up with the idea that this reminded him of a sorting out of combinations. And I'm going to say a little bit more about that in a moment. But what is quite interesting, and I only discovered this a few years ago, Charles Darwin, more of him later, also carried out some crosses with plants, in his case snapdragons, also recorded these 3:1 ratios, exactly the same time, 1868, but left it there. He just recorded it, published it, you can see it in one of his books, but he didn't try to push it further like Mendel did in his paper of 1865. Well, seeing these simple numbers, ratios, 3:1 and so on, led him to propose what is essentially a particulate theory of heredity. That's the key point, a particulate theory of heredity. By particulate, what I mean is really two concepts, that there is a particle which is unchanging, and those particles are being passed on from generation to generation. So unchanging particles are being passed on. We would now call them genes, and they play an important role in determining differences between characters. He then proposed, to get the numbers right, that in each plant there were two particles that were responsible for the character or the character differences that he was interested in, but only one of those two particles would be passed on in the gametes that made, whether that's pollen or eggs. But on fertilization, on pollination, what happened is that you would have one particle from one parent and one from the other, producing the two particles that you see in the adult plant. And he postulated that one particle might dominate over the other in determining a character. And if you put all of those things together, you can then explain the sort of ratios that you get. And as I said, I'm not doing Genetics 101, so I'm not going to do that, but I'm sure you can get a sense of where the logic was going. Also implicit in that the reasoning is that these particles contain a principle of information that's passed on. That's the only way it could actually work. He's not explicit about that. That occurred some years later, but that is obviously implicit for these ideas to make sense. Now, these segregations, I've just tried to illustrate here the sort of 3:1 ratio that I'm talking about. This was, frankly, a brilliant piece of abstract reasoning. I mean, it was really brilliant to make sense of this, to do these experiments, to come up with what was essentially exactly right. He had lots and lots of good data to support his ideas. In fact, the data, as Fisher, the famous British statistician in the 20th century, analyzed his data and concluded that it was too good to be true, and therefore that it was, there must have been some cheating going on. And of course, Fisher blamed the gardener rather than Mendel himself, saying that the gardener knew what Mendel wanted to find, so he'd throw in a few more seeds in, one category or the other. I have no comment on this. But it was a brilliant piece of abstract reasoning, absolutely right, but nobody took a blind bit of notice of it. And it wasn't that it was hidden away in some gardening journal in Brno, it was in a journal that was widely read. And it was even reported, I've discovered by accident, in the Encyclopedia Britannica of the 1880s. So it was well established. But nobody took a blind bit of notice of his models, okay? Absolutely no of his model. However, 35 years later, just at the turn of the 20th century, 3 different sets of investigators did the same types of experiments and came to the same conclusions, began to write up their work, and discovered they'd been scooped by the gardening monk, who by then was dead, 35 years previously. The first group that tried to publish the data, I think, did their best not to refer to Mendel, but the second group, who realized they'd been beaten by the first group, dug around and discovered it had been discovered by Mendel, so they did refer to it. And in the end, it all sort of came out that they all ended up having to refer to Mendel. Now, why was it took 35 years for this to be taken notice of? I don't, of course, know, but between 1865 and the early 1900s, chromosomes have been discovered. Here's an early 19th century picture of a squashed onion trip, and you can see in here chromosomes like here separating as the cell divides, and it was being speculated that these chromosomes might be carriers of hereditary determinants. I mean, just in a general sense. And then it was discovered by another investigator called Van Beneden, working with little worms, with little nematodes, which happened to have few chromosomes. And this is another example of choosing the right material to study. This nematode only has 4 chromosomes, and when it makes gametes, egg and sperm, you only find 2 of those chromosomes in the gametes. And this is the reductional division that Mendel had proposed to happen every time you produced, in his case, pollen and eggs. And what I think is striking about this is that Mendel's rather abstract thinking here had been translated to a sort of concrete basis. And scientists, and maybe biologists in particular, are more at ease when there's a concrete basis for something than just abstract reasoning. Physicists and so on tend to be happy with the abstract reasoning. But here, I think when there was something that behaved like the abstract reasoning predicted, it became a much more powerful support for the original idea. So Mendelism was born, the science of genetics was born in the first decade of the 20th century, and I think it would be fair to say that genetics was one of the major forces of biology in the 20th century. It took a little while to translate genetics into molecular genetics. First of all, DNA had to be discovered as the hereditary material. That's usually associated with Crick and Watson, but that actual discovery was not made by Crick and Watson. It was made by Avery, McLeod, and McCarty, I have to say at Rockefeller University, where I was working till earlier this year. And they discovered that by studying pneumococcus, a bacterium which it can grow up in a variety of forms, but they looked at a form which makes rough colonies and smooth colonies, and they found that if they took extracts from one colony form and added it to the other under certain conditions, then it could turn the other colony into the one where the extract was made from. So there was what they called a transforming principle, and they did very careful biochemical fractionation and came to the conclusion that the chemical component that was transforming the pneumococcus was deoxyribonucleic acid. This was very contentious at the time, not least of all by some of their colleagues at Rockefeller University. They had their fiercest critics were their immediate colleagues, because DNA was such a simple molecule, or thought to be, compared with proteins, that it was difficult to see how information could be encoded in that. And that required the structure of the DNA, which of course was Crick and Watson, based on the experimental work of Wilkins and Franklin, and they produced the model, which I'll refer to again somewhat later, of the double helix, being made up of a series of nucleotides, and the order and type of nucleotides being a code, it's the best way to think about it, for the information that makes up the gene. So those chromosomes that I showed you, when unraveled, are made up of DNA, the double helix of DNA, and this could be easily copied because these are complementarily paired, and so if you pull them apart like a ladder, you will make a complementary copy of it. Crick, Francis Crick, who, after which we've named the new institute we're setting up in London, went on also to play a major role in cracking the genetic code and proposing what we call the central dogma in molecular biology, where information encoded in the sequence of DNA is transcribed into the sequence of RNA, that's ribonucleic acid, very closely related to DNA, but just less stable, and then that is translated into a polypeptide chain shown here of amino acids, and those amino acids, shown here, then determine the character and order of those amino acids, determine the property of the proteins that are made, which determine many of the characters in cells. So here you have a molecular explanation for the data of Mendel, and here you have the concept of the gene laid out. So this is my second idea, the gene, very powerful idea, the basis of heredity. You can explain genotype to phenotype, by which we mean genotype is the genetic makeup of an organism, the phenotype is the characters that are expressed, and we see that through the central dogma. Genetics, of course, is also very topical and important for us today. For many, many reasons, I put one up there, including implications for what we are as human beings, by which I mean how much are we determined by the genes we inherit from our parents, how much from environment, what does it mean for us, what does it mean for responsibility in criminal codes. All of these sorts of issues are still very much tied up with genetics. It's a very, very topical subject. So that's my second idea. Well, my third idea. You knew this had to come up. Evolution by natural selection. You would be forgiven if you go into a bookshop and look for the popular science shelves, that there are actually only two topics in science. One of them is string theory, and the second is evolution by natural selection, or evolution in general terms. It is something that is found very fascinating, both of those topics actually, and for some, it's contentious. Now, this is actually a simple idea, but a very powerful and beautiful idea, probably the most beautiful idea we have in biology, but it's actually very straightforward, and it is central to all biology. Now, it's an idea that has two parts. The first part of it is that life evolves, that is, it changes over time, and therefore there is a tree of life with all organisms being related by descent. You can draw a tree of how living things are related one to another. So that's the first idea, the first part, that life evolves. The second part is natural selection, and here the notion is that the major mechanism for that evolution is natural selection. Now, both of these ideas are usually associated with Charles Darwin, who's shown here on the right. But in fact, the first of those ideas, that is that life evolves, was absolutely not Charles's idea. I'll say something about natural selection and how unique that was for Charles in a moment. But the first idea that life evolves was discussed very much in the 18th century by French scientists such as Buffon and Lamarck, but in particular by Charles Darwin's grandfather, Erasmus Darwin. And Erasmus Darwin is here on the left. And I want to say a few words about Erasmus Darwin because he's one of these heroes of British science that virtually nobody has heard of. He's also actually quite an entertaining and colourful character, rather more so than Charles himself. So Erasmus, who lived in the latter part of the 18th century, was a doctor, an MD. He worked in Lichfield and later in his life in Derby, and he was a member of the Lunar Society, which some of you may have heard of, which is a society of scientists and technologists and entrepreneurs who used to all get together to discuss scientific problems. Very important at the birth of the Industrial Revolution. I actually think if scientists, technologists and entrepreneurs got together a bit more often now, and we wouldn't be in quite such a mess as we are now. It's all sort of segregated out into, and we're just run by the financiers, which I don't think is good at all. But anyway, the Lunar Society would meet once a month under the full moon. They were called the lunatics for that reason, because of their odd behavior. They could ride home on a horse in the full moon, probably a little inebriated, I imagine. And they would discuss scientific topics of the day. And the founding member, one of the founding members, was Erasmus Darwin. He was a doctor, but he was also a very charming man, a conversationalist, though he had a deep stutter. He was very fat, and he cut out of his dining table a sort of half oval so he could sit nearer to his food. He was famous as a doctor, not because he actually cured many people. I have to be quite honest with you, doctors didn't cure many people until about 1900, really. But mainly because he was very good at telling you whether you would die or not. And that was quite useful, because if you knew you were about to die, then you could put your house in order. You could perhaps, write that will that you were too nervous to write before that because who you might upset and all those sorts of things. And he was obviously a very good observationalist. He just didn't know how to intervene to cure people. He charged according to income, so he made all his money from rich patients. It's rumoured that George III wanted him to be his physician. He didn't take that up. He was a closet Republican. But he also was a scientist, and he was a poet. And for some years, he was the most best-known poet in England. I mean, it's quite extraordinary. And he wrote most of his scientific work up in the form of blank verse. And I have a number of the 18th century editions of his work, and you read this stuff, I mean, it's just hilarious. He'll be talking about the sex life of plants, shall we say, and referring to Cupid and so on, and at the same time discussing the crosses that were going on and what might be happening. He also was a proponent of female public education and wrote a book on that and set his two daughters up as teachers in the school based on the principles. Anyway, he argued that life evolved. It was completely clear that he thought life evolved. And he put his motto on his coach, which was, in Latin, of course, everything from shells. The idea being that shells, if you open up a shell, the mollusk is a formless mass. And he argued that everything evolved from such simple life forms. This led to him being considered a dangerous character by the dean of Lichfield Cathedral, and he lived in Cathedral Close, who told his patients not to go to him, and he had to paint out his motto because he earned all his money from his rich patients. He's an extremely interesting person. If I've whetted your appetite, Desmond King-Helly, who's a fellow of the Royal Society, actually, who was interested in artificial satellites, has written a number of books about Erasmus Darwin and also the Romantic poet Shelley, for example. But the point was not that this idea was around, it was the idea of evolution, but the evidence, the data for it was not really very solid. And that's where Charles comes in, a younger version over there on the right, who collected vast amounts of data supporting the idea of evolution based both on the fossil record and on living animals and plants. And this came about because Charles went on a trip in the Beagle. You're probably familiar with this. Great story for 5 years. He then retired to Down House on the outskirts of London in Kent, thought about this problem for many, many years, and produced The Origin of Species in 1859. He reported in a paper in 1858, but reported The Origin of Species in 1859. In The Origin of Species, there is only one illustration. It's actually beautifully written, but there's only one illustration. And that is a tree of life that you see here, where he connects all living things. There's extinction going on here. And at the top of the species, this is the first tree of life drawn in this way. Notice he didn't quite know whether life arose in more than once. So at the bottom there, he sort of left it open. He doesn't quite know about this. And he had produced evidence that evolution had occurred, but he did more than that. He also produced an idea, that is, that this occurred by natural selection. And he was stimulated to think of this during his South American trips on the Beagle, particularly when he visited the Galapagos, which I recommend to all of you, where he observed that the Galapagos Islands are new islands that arose volcanically. They were colonized by, or are thought to be colonized by a finch, and that finch then diversified over 15 million years or something of that sort, until all the different habitats and lifestyles are inhabited by closely related but different types of finches. And by studying their beaks, he came to the conclusion, and you can see from the left here, that they were exquisitely adapted to what they had to do, whether it was cracking nuts or biting or catching insects or whatever. And they were beautifully adapted to do that. And as a metaphor for that, I put up these sort of different pincers and pliers just to show how the same basic principle can be adapted according to the job that you actually want it to do. And that's what he observed. And this led him to the idea of natural selection. Now, there's an interesting story about his publishing that idea, because he sat on it for 15 years. Whether accumulating data or whether because he didn't want to, because he was embarrassed about making the case that, that evolution was taking place because of the implications for the origins of mankind, people don't quite know. But he was stimulated to go public when Alfred Wallace, another English naturalist who was suffering from a malarial fever in the Indonesian peninsula, had exactly the same idea, probably under the influence of the malarial fever, in actual fact. And he wrote a letter with a paper in it to Darwin and said, I've had this idea about natural selection, what do you think of it? And this arrived in Charles's, in Down House, and suddenly he saw that somebody else had had exactly the same idea of him. It was beautifully argued, nothing like the amount of data, but the same idea. He didn't know what to do. He contacted Hooker, who was then the director of Kew Gardens, and Hooker had the idea that both papers, one from Wallace and one from Darwin, should be read at the same time in 1858 in the Linnaean Society. Now, the Linnaean Society, you may not know this, was actually in Burlington House, where the Royal Academy of Arts is in London, just off Piccadilly. And if you go in there, there is a room called the Darwin and Wallace Room, which is the exact room where these two papers were actually read out. So it's something you can actually go and visit. Nobody took too much notice of this, it has to be said yet again, but it stimulated Darwin to publish his book, the great book, The Origin of Species, in 1859. Once he'd published it, though, he got a letter, and this you may not be aware of, you may be aware of the Wallace story, from somebody called Patrick Matthew, who was a tree grower. And Patrick Matthew had written a book on the growth of naval timber in 1831, because timber for boats was very important just post-Napoleonic times. And he has a page describing the concept of natural selection. And that was published in 1831, so a complete precursor. So even the idea of natural selection was not actually, strictly speaking, Darwin's alone. Matthew, who wasn't upset at all that Darwin got all the credit, produced no evidence for it, didn't extend the idea, it was very limited, but the idea is very clear. So what is the idea of natural selection? It is, quickly stated, that within a species there are variants, and these variants are due to inheritable differences to the genes that we were talking about with Mendel, though this was not known at the time. And what is argued here is those variants that are most successful, and by most successful, what is meant is they reproduce more, and therefore pass on more of their genes to the next generation, will tend to inherit more of the genes that are responsible for the characters that make them successful. This is so-called survival of the fittest, though that's not Darwin's term, it's Spencer's term. And that the gradual changes within a population in the gene pool, in the genes that are being inherited, will gradually lead to changes in the characteristics of that species, until eventually you will get speciation, and therefore a new species, and that is the basis of evolution. It is actually a beautiful idea, but very simply stated. Now, I want to slightly repeat that argument, linking it to the two earlier ideas that I've mentioned to you, because, and the reason, the cell and the gene, and the reason I want to do that is that a geneticist called Muller in the 20th century argued that he thought this concept of evolution by natural selection was so important that you could define whether something was living or not as to whether they had properties which allow them to undergo natural selection and therefore to evolve. that's not a description of the object, the entity. It's a description of the historical process that led to the entity, and he felt that was the fundamental principle. And I just want to show you that evolution by natural selection will fall out of the fact that if you have a cell and it has a heredity, a heritable component, the genes, then it will work. And those properties that Muller was referring to is that life must reproduce, which of course it does. Life must have a hereditary system whereby information defining the characteristics of the organism is inherited during that reproduction. And thirdly, that hereditary system exhibits variability upon which natural selection can work. And that I'm just showing you here in this slide, where we have a cell. That's the idea of the cell. Within it, we have the genes, which determine the characteristics of the cell. And the only characteristic I'm interested in is orange and red outer skin of the cell. And what you see there is that every time the cell reproduces, which is the first of Muller's statements, they also copy their hereditary component. That's the double helix here. Within that are genes that determine the characteristics of the cell, whether they have, for example, an orange coat or a red coat, that there is variability either due to errors in copying or to mutation. And so in these two lineages, there is a mistake in one of the genes on the right that leads to a red shell as on the cell instead of an orange one. And if that's advantageous in some way, like it divides quicker or doesn't get eaten so quickly, then that will tend to take over the population. So what this means is these three ideas I've talked about are linked together in a sort of triangle supporting biology. The cell and the gene will give rise to evolution by natural selection, and that will be inevitable based on that concept. And I think that there are three ideas that we need to hang on to. Well, Charles became very famous. This is what the iconic image we sort of have of him. And this is the last sentences in The Origin of the Species, actually, which I love to read out, I have to say, because he's quite poetic. Whilst this planet has gone cycling on according to the fixed law of gravity, notice how he links himself with Isaac Newton, you know, quite different. And then he says, to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So he really wants to place his idea in biology in the same place as Newtonian physics had, Newton had in physics. Well, this is still very important to us. The evolution by natural selection, the third idea, all life evolves, it's related by descent. Natural selection, major mechanism, there are other mechanisms that are important. And a couple of things I'd like to say. Kin selection is relevant to our thinking about altruism and maybe morality. What do I mean by kin selection? It's simple that if what matters are the genes that are being inherited from generation to generation, then since you are related to your kin, it would help the transmission of your genes if you were to help your kin reproduce. And what it sort of means in its crudest term is that it's worth sacrificing yourself for two of your siblings, and three definitely, because then you'd pass on more genes to the next generation that you have. And although it sounds a little odd, that altruistic behavior has a genetic basis and maybe should influence our thinking about altruism and morality. And the fourth point I really like is that the tree of life, the fact that we're all connected, means, of course, that we are related to every living thing in the biosphere. And since we are supposed to be, at least at the top of this biosphere, you could argue as a consequence of being related to every living thing, we have a responsibility for our relatives, a responsibility to be good stewards for our relatives. And that's the best argument that I can think of for why we should take care of the biosphere. So those are the three ideas. I want to move to a fourth, because certainly the idea of evolution by natural selection is essentially an historical idea. It tells you how life might evolve. It doesn't tell you how life originated, which is a different issue. But somehow it doesn't give you a feeling for what life is. And for that, I'm going to move to the fourth and then eventually to the fifth idea. The fourth being the idea that life can be understood in terms of chemistry and physics, the mechanistic basis of life. And the reason I want to emphasize that is because it was not at all obvious for centuries that that necessarily would be the case. Aristotle, for example, argued that living things had a natural end or goal, and movement towards that teleology, that is a goal, was driven by an inner vital force, to quote him, and that was the origins of what was known as vitalism. That is, that living things are not based on normal physics and chemistry, but are something different. And the replacement of the idea of vitalism, that life is best understood in terms of chemistry and physics, forms this fourth idea. And central to this idea, curiously, comes initially from thinking about the chemical changes that occur during the fermentation of fruits. And it has its origins with the great chemist Lavoisier, and this is a painting done by David. It used to belong to Rockefeller University. It used to hang in the president's office. And they, during one of their periodic funding crises in the 70s, they sold it. It's now worth $100 million, would you believe? They sold it to the Metropolitan Museum of Art, where you can see it. And you see he's there with his laboratory assistant, who was also his wife, who survived him and went on to marry Count Rumford later in her life. Now, what Lavoisier did, working particularly with Laplace, in actual fact, looking at the chemistry of life, he studied a guinea pig undergoing respiration and compared it with burning charcoal. And he came to the conclusion that there was a close relationship between the amount of heat produced by a respiring guinea pig and by a burning charcoal and the amount of carbon dioxide that was being made. And he thought there was something very fundamental about this connection, that there were chemical reactions going on within the guinea pig that were producing the carbon dioxide that were related to that. And he didn't get very much further with this because, unfortunately, Lavoisier had been a tax collector, and during the Revolution, he was arrested. Here's another picture of Lavoisier. In the background, you'll see the guillotine. He was arrested, and despite being actually a big supporter of the Revolution, he was tried in 1794, and the judge said, and this is, I think, something that all scientists need to think of today when they start meddling with politicians. The judge says, "The Republic has no need of chemists and savants." The Republic has no need of chemists and savants, and he was guillotined later in the day. Well, it was taken up, the work he did, by Louis Pasteur, and he demonstrated that living microbes were responsible for the chemical transformations that occur in fermentation. He studied beet sugar fermentations in Lille, which was going bad. It went sour. And he showed that when it went sour, it made acid instead of alcohol. He looked at the fermentations under the microscope, and he found that the acidic ones had bacteria in them, and the good ones had yeast in them. And so he could advise, this is a good bit of translational research, with impact, he could advise the beet sugar fermenters that the way they could get decent fermentation was to put yeast in it. But of course, he then made the scientific conclusion, which was this, in French, but translated, fermentation was a physiological act yielding chemical products necessary for the cell. That's the conclusion. And then he concluded, such chemical reactions should be thought of as an expression of the life of the cell, clearly stating chemistry was the basis of life. Well, this is central, this idea, to our understanding of biochemistry. It's really the birth of biochemistry. And it's really the chemistry in cells is based on these. This is a protein molecule, and the protein has different proteins have different shapes and different charged surfaces and can like water or dislike water, and those surfaces and changes produce chemical microenvironments that can catalyze chemical changes. So they are catalysts that can work in very, very gentle conditions. And I just think about them sort of changing the shape of the substrate and causing chemical changes. But what's interesting in living things is that this is a metabolic map, and each one of those dots is a chemical change, an enzymatic action. And you'll see hundreds, if not thousands, of chemical reactions are going on in the cell at the same time. It's completely extraordinary. And many of these different reactions, for us to do it, we'd have to heat to high temperature or treat with acid, and it's all occurring in these very simple ways within the cell. And I like to think of the structure of a cell. This is an ideal form of a cell, not simply in terms of structure, but all of this is providing different microchemical environments to allow many, many reactions to going on at the same time. It's a different way to think about what cells look like. It also requires for these to be connected to each other, which we'll come to in the final idea in a moment. So that's how I like to think about the cell. So this is my fourth idea. Life as chemistry. It provides a mechanistic explanation of life. Primarily, we think of chemistry. There's also physics here as well, which is important, but I haven't emphasized that. But this is the basis for understanding many diseases, the chemical basis for disease. It's central to medicine and the pharmaceutical industry. When you take a pill, you're taking a pill that interferes with the chemistry of your body to correct something or to kill a bacterium or something, but it's based on the fact that life is chemistry. So this killed vitalism and is crucial to the health industry and underpins molecular biology as well. These four ideas, I think most biologists would agree with. The fifth idea, I'm not sure everybody would agree with, and it's my last idea. And if you're getting a little tired and thinking I'm talking a bit too long, I apologize for that, but I suggest you take one very deep breath because we're on the final straight, okay? But there are some bits in this which are a bit difficult, okay? But, you know. So this idea is to think of biology as an organized system. An organized system based on information and the management of information. And the reason I'm proposing this is that it sort of builds on ideas that are current around complex and systems theory and systems biology. Now, although this is current at the moment, I've got to say the ideas have been around for years, many years. I even found that Immanuel Kant, the philosopher from 1800, wrote about 20 pages about thinking of biology as a complex system in a book on moral philosophy. I haven't got the slightest idea why he thought it was relevant to moral philosophy, but it's certainly not an idea that has just come out recently. But thinking of complex systems is important in a number of studies outside biology to do with weather, with social structure, with transportation networks and the like. Now, in biology, our idea is to try and explain higher order biological phenomena. That's what we're trying to do. Phenomena like homeostasis, communication, spatial and temporal organization. And what I think we have to do here is we have to find effective ways of translating the descriptions of the chemical and physical processes of life into the management of information. I mean, we start with physical descriptions, but to understand it, we have to think about how the information is being managed. And by management of information, I mean how you gather information, you store it, process it, how you determine outputs based upon the inputs that are going in. And this sort of thinking emphasizes regulation, the understanding and structure of networks, of systems, of so-called emergent behaviors, all these sorts of phenomena. Now, to illustrate the point, I'm going to give you two well-known examples, which are known to all of you, just to get a sense of the thinking. The first of these is, I've mentioned already, the structure of DNA. Now, this is the double helix. It's been copied here, unraveled and copied. I talked briefly about this before. Now, this is the iconic image of biology from the 20th century. It's it's a beautiful idea of the double helix. But the beauty is not really in the structure. The beauty is in the idea that is encapsulated in the structure. Because when stripped away, what this is a digital information storage device. That's the way you understand what it is. Encoded in here in the nucleotide sequence is digital information, and that's the basis of heredity. Now, the fact is, just understanding the chemical structure does not inform the biology. What informs the biology is understanding what that structure means for information. And in this case, it's the storage of information, which is the basis of heredity. And it's only when the chemical structure is expressed in terms of information does it make biological sense. And that's why I'm putting so much emphasis on that. The translation into information management makes biological sense. The second one I want to draw your attention to is metabolic regulation, in particular regulation of bacterial transcription by Jacob and Monod. This is a picture of a governor from a steam engine I discovered in a working boat in New Zealand. It was made in the 1880s, 1890s. You know, there's one thing, I don't know if you're the same as me, I sort of understand all machines built before about 1900 and virtually no machines built after 1900. So I'm constantly forced to sort of look at metaphors in objects built before 1900. In this particular case, it's a governor, and it's a steam engine and it spins. As it spins, the balls are moved out by centrifugal force. That lifts up a valve and stops steam going into the engine and slows it down. So it's in fact essentially a negative feedback loop of the sort that we have up here. This is translated into a biological system, and you have an enzyme going from A to B to C, 3 enzymes catalyzing these reactions, and C feeds back on the enzyme going from A to B, shuts it off, and so it's homeostatic. Now, these ideas were first clearly stated in the biological system by Jacob and Monod, who was looking at transcription of a gene called the lac operon in bacteria in Escherichia coli, which catabolizes sugar. Now, once again, you could describe that network in terms of the chemistry. You have a protein, you have DNA, these touch each other, you have small molecules, and you can just describe it, but you only understand it when you put it in the form of a negative feedback loop, because that's what it's doing. When you follow the information and work out what's happening with the information, then you understand what's going on. And of course, this is a negative feedback loop. I've put a positive feedback loop here, which, as you see, will turn this whole thing into a switch. Once you make C, you make more of C, and you drive it into an actual switch. And so if you sort of take this further, then you could imagine all the processes going on in cells and organisms in terms of regulatory networks. And an analog of that would be, for example, an electronic circuit, where you have all components managing the electricity going around a circuit, but they're basically managing information. And what's been put in here is different gene names to sort of indicate that as a metaphor. But in here, and if it was a computer, you have the hardware, and we'd have the software we'd put in it. Interesting in biology, which has got the same concept, the hardware, the wires between the different components is through diffusible chemicals. This has been a point made by Dennis Bray. So the hardware can be easily rewired. Simply by the diffusing chemicals going from one component to another. And so in life you have a richer behavior. Not only can you reprogram through what you encode in the DNA, but you can also rewire the hardware which you should consider as wetware, in actual fact, because of that. So here we have sort of an interesting way, and I'm going to finish with 1 or 2 examples of how this type of thinking may illuminate the problems that we have. Because what I'm arguing here is that we need to consider living organisms as a complex chemical system, wetware, acting as a logical computational machine made up of linked logical modules. That's the essential basis here. And we can learn from other systems. We can learn from, in this case, transport hubs. Because if you've got a complex network, you can map how the different components in that network are linked together. And in this case, what you will see, this is a transport map, is that certain hubs are important and others are not. So in this case, Houston, I think this is probably Houston down here, is crucial. If you knock out Houston, the whole system breaks, whereas we don't care at all about Kansas City, for example. You can take Kansas City out and it really doesn't matter. Now, it turns out that the way that networks are put together in efficient aeroplane route hubs have some relationships to the way networks are put together in living systems. And there's things to be learnt from that and social networks as well. So that's one bit of thinking that is sort of quite interesting. Another thing that we need to be aware of is that pathways, networks, are not necessarily as simple as we'd like them to be. We tend to think of pathways as like linear pathways where you start something, you go to an end here. But the reality is rather more complicated. So intuitively, you'd know if you switch this on and off what would happen here, but if you switch this on and off, it would be very difficult to sort of quite know what it's doing. And this is actually what life is like. It's much more complex. It's not so linear. Now, we have a bit of a psychological problem here, in my view, because we tend to think much more clearly like this. We don't do very well with this. And as a consequence, we try to force our interpretations and our experiments into this type of thinking because we can be comfortable with it. Where if the reality is like this, we're going to disturb what we're doing. So we have to accommodate complex networks. And different types of people think more in one direction than the other. And I found this following picture, which I never would dare to show in the United States, in a pilot's manual, okay? Now, I dare show it here, okay? The notion is men think like this, and they basically are on or off, and women are altogether more sophisticated in their thinking and more complicated. And I would like to suggest that we need to think much more like women in solving these problems, which is a good reason why we need a good mix of people. Now, a final point about networks, and that is that if you look at a signaling network, which is just working through here, then this is a simple system. You switch this on and you switch this on. If you switch this off, you switch that off, okay? And so that's very straightforward. It's like a traffic light or something, you know, an on-off switch. But if you now introduce the concept that dynamics are important, then you can pulse information down here, and pulsing information can convey much more information in the same hardware, because this message of a pulse going down there in time can mean something quite different from this. Now, this is inherent in most biological signaling systems, but we've only just begun to scratch the surface that dynamics matters. And it matters a great deal, and I'll just demonstrate it to you in this metaphor here. If you just have an on-off switch, you have a light switch. If you now have dynamics, an analog of that is, of course, the Morse code. And you move from just having a yes-no to being able to write the works of Shakespeare. I mean, it's an incredibly information-rich transition if you introduce dynamics. I'm not suggesting that the cell is writing works of Shakespeare. I'm only saying thinking about it in that way is important. Well, we've got to make structures. I'm getting late. I won't talk about it. That's a simple structure. Thinking about Turing and others might help us with that. But I do want to finish with just a brief discussion about the complications that we have by talking about Harrison's clocks, okay? Because one of the problems we have in thinking about complex systems is if we think that they are intelligently designed. I use my words very carefully. If we think they are intelligently designed, we run into trouble. Because they are not intelligently designed. They've emerged through evolution by natural selection. And why is that relevant here? Well, it's relevant because I'll tell you what happened with John Harrison's clocks, which were intelligently designed. And these were designed to solve the longitude problem. You can read Darby-Sabel if you're interested in that. And basically, you had to make a very accurate clock. This is work from the 18th century if you wanted to determine longitude. And Harrison started by making this clock, tested it, and it didn't quite work accurately enough, thought about it, made this one over here, completely new, thought about it, made this one here, still didn't quite work, ended up with this one, okay, which worked beautifully. Now, the point about this is, at each stage, this did not evolve into that, into that, into that. He went back to the drawing board and intelligently designed it. What we have within us are the vestiges of 3.5 billion years of evolution. And we have all sorts of junk around that happens to be there because we inherited it. And what we shouldn't do is try and invest a lot of significance in that junk. Okay? We have the appendix, which is just one typical case of it. But this makes it difficult for us to do. And one of the reasons the networks are so complicated is because there's lots of redundancy in there, and it makes it very difficult to actually work. So if we are going to try and analyze complexity in biological systems, then I think we may end up in a slightly stranger world, because thinking about complexity leads to counterintuitive conclusions. And biological explanations up until now have essentially inhabited a common sense world. You know, when I think about an experiment, I sort of imagine my cell here, and I imagine it in this room, and I've got this touching this and so on. It's Newtonian. It's very simple and straightforward. But I wonder, just like physics did at the beginning of the 20th century, whether we may move into a stranger world with this notion in biology. Because up until 1900, it's sort of all as common sense. Then you had relativity. And you know what relativity is like. You read a book about relativity, you think you've got it, you know, you're sitting on a beam of light, traveling at the speed of light, you're sort of almost there, you shut it, and then it sort of fades away, you know? And the reason it fades away is because it's not part of our common sense world, you know? It all makes sense, but it sort of only makes sense in this sort of strange logic and to some extent mathematics. And if you think you've got trouble with relativity, then move on 25 years and get into quantum mechanics, and you're completely in la-la land. I mean, it simply makes no sense at all in terms of a common sense world. And what I'm saying is that maybe in biology, because of the complexity and the complex systems there, we may move into a stranger world where we can't operate as if we're in a common sense world, and we may need to borrow a lot from the physicists, not explicitly in what they've been doing, but the type of thinking that they've been used to for nearly a century now, which biologists have not. So these are my ideas. Now they've become the great ideas of biology. So it's morphed during the course of the evening. And we have the cell, the gene, evolution by natural selection, life as chemistry. I think we'd all agree on those, and maybe biology as an organized system. Thank you very much.